My name is Nur Shakira binti Shamsuddin and these are my team members. I will start with the introduction for our field study analysis. Coleoptera, commonly known as beetles, are the largest group of organisms. The most notable characteristic of beetles is the transformation of their forewings into elytra, a hardened sheath. Coleoptera are the only order of insects that have elytra. Their scientific name derived from their adaptation, which is coleos mean sheath and petron mean wing. This adaptation has enabled them to expand into many habitats such as leaf litter, logs, and soil. However, the order Coleoptera can also be found inside our house. After several days of observations, we found that the bushes in front of the Pusat Pentadbiran, Faculty Applied Science Building, and around College Gamma B at UiTM Tapah are common habitats for ladybugs and other types of Coleoptera. Therefore, the objectives for our field study analysis are: number 1, to study the diversity of Coleoptera species in various habitats within UiTM Tapah, Perak, and number 2, to identify suitable techniques for capturing Coleoptera for field study analysis at UiTM Tapah, Perak.
So now we'll move on to the methods that has been used during this field study. There are 3 methods, which are hand-picked, sweep net method, and pitfall trap. For hand-picked, we choose this method because most of beetles are easily to be captured and they has low rate of hop. However, we still use glove in advance as for precaution. Next, we use sweep net method as some of the species are hard to visibly found. So by sweeping the net on the ground, it can help by reveal them, by triggering them, and capture the beetles. Lastly, we use a pitfall trap as to lure on the ground beetle that are hard to casually found by embedded a plastic cup with a beak in it. Once the beetle has fallen into the trap, it shall not easily escape as the smoothness of the plastic cup surface. After a few hours, the trap shall be checked again if it has got any into the trap. However, we found that this method are not suitable at the moment because it has been raining cat and dogs during the searching time, causing the trap to be flooded with the rainwater and fail. And here are the clips of how these methods are done.
This ain't Texas. Ain't no hold 'em. So lay your cards down, 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 down. So park your Lexus and throw your keys up. Stick around, round, 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 round. Stick around. And I'll be damned if I can't slow dance with you. Come pour some sugar on me, honey, too. It's a real live boogie and a real live hold down. Don't be a bitch, come take it to the floor now. Woo! There's a tornado, in my city, in the basement. That shit ain't pretty. Rugged whiskey, we surviving. Of red cup kisses, sweet redemption, passing time, yeah. Ooh, one step to the right, we headed to the dive bar we always thought was nice. To the left, then spin me in the middle, boy, I can't read your mind. This ain't Texas, ain't no hold 'em. Lay your cards down, 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 down. So park your Lexus and throw your keys up. Stick around, round, round.
We conducted our first visit to the 3 selected locations, which are bushes in front of the Pusat Pentadbiran, around College Gamma B, and the Faculty Applied Science Building. The exact time were 8 a.m. until 10 a.m. in the morning. The species that we found were 13 Coccinella, 10 Adoratus, 6 Vilophaga, 2 Anomela cuprea, and lastly 7 Tenea Bryonidae. For our second visit, we conducted a week after the first visit, at the same time, which is in the morning. The species we found were 10 Coccinella, 7 Adoratus, 8 Vilophaga, 2 Anomela cuprea, and lastly 5 Tenea Bryonidae. Our findings on the second visit are less than the first visit. This is because the second visit was conducted on a rainy day, which made the species of Coleoptera hide to find shelter and avoid from the rainwater. 5 species of Coleoptera were found during this study field. The first species are ladybugs. The scientific name for ladybugs is Coccinella septempunctata. They live in temperate climates that can be found almost everywhere, such as gardens and wheat patches. Ladybugs' unique characteristics are they have a hard shell with a dome shape to protect their body and comes in bright colors like red with black dots. These characteristics warn their predators that they are toxic or distasteful. They are active hunters that eat small arthropods like aphids and mites. The second species is the Chinese rose beetle, or their scientific name is Adoratus sinicus. They live in warm and humid climates for their survival. Their characteristic is they have a glossy brown coloration. The Chinese rose beetle are active hunters and feed on plant foliage of many species that can destroy the plant. The next species is the May beetle, or their scientific name is Philophaga. They live in grassy areas, lawns, and gardens. They have unique characteristics which are black or brown body without patterns and club antenna with segments. They are similar to the Chinese rose beetle as they are nocturnals. They are active at night and feed on plant leaves or flowers.
Fourth species are Cupreus scheffer, which are known by the scientific name is Anomala cuprea. They are frequently found near the pods as they prefer living in humid and low temperature environments. Their unique characteristic is where they have shiny green blue body that act as their defense mechanism as to disguise in the environment. And also it has white legs for fast movements. It is a nocturnal scarab beetle, which means they are active at night and feeds on flowers and leaves. And lastly, we have the darkling beetle, which came with the scientific name of Tenebraeunidae. They are commonly found on the ground, under or around the rocks, logs, and leaf litters as they love darkness. Their unique characteristic is where they have brown and black body, segmented antennae to gather information about their environment, notched eyes, and segmented legs. They also feed very widely, such as dead animals, including other insects, fungi, and plants.
The diversity of Coleoptera plays an important role in the ecosystem. The first importance is they can be a natural predators for the pests that usually attack and destroy the plants. By using species in Coleoptera, the population of pests can be controlled. At the same time, the usage of chemicals in pesticide can be reduced for agricultural purposes. For example, ladybugs become a natural predators to control the population of aphids in agricultural area. The next important are the species of Coleoptera can be an important pollinator for some specific plants. For example, some species of beetle can be an important pollinators for some specific plants. This interaction contributes to the reproduction of the plants and overall of the biodiversity of ecosystem. The last importance of having the species of Coleoptera is they can act as a bioindicator to detect the quality or changes in the environment. Since ladybugs and beetles are sensitive to their changes of environment, the presence of them can indicate a healthy and diverse ecosystem.
We move to the recommendation. The first one is study interaction. We need to investigate the ecological interaction between Coleoptera species and the environment, including predation, competition, and symbiotic relationship. Second is seasonal pattern. We can investigate seasonal pattern in Coleoptera activities such as emergence, reproduction, and feeding behavior to gain insights in their life cycle and ecological roles. And the last one is utilize technology. Incorporate with technologies such as remote sensing, DNA barcoding, and GIS mapping to enhance data collection, analysis, and visualization in the field study. In conclusion, the field study analysis of Coleoptera species around UiTM Tapa area provided significant findings about the diversity, distribution, and adaptation of these beetles in their natural habitats. Through systematic observation and data collection, the study revealed the richness of Coleoptera species presence in different habitats. The findings highlighted the importance of conservation efforts to protect these insects and their habitats.
Additionally, the study identified suitable technique for capturing Coleoptera for field research, enhancing our understanding of their behavior and ecology. Overall, the research contributed to the body of knowledge on Coleoptera species in the UiTM Tapa region and underscore the significance of preserving biodiversity for future generations. Here we provide our reference. That's all from us. Thank you.